So, good afternoon, everybody, and welcome to the last but one talk of this highlight track session for today. As you can see, during this presentation, I would like to demonstrate how we can dynamically model cellular decision making. Okay. So, cells need to take decisions in several biological processes. For example, cells need to decide whether or not to grow in size during cell division or cell proliferation, or whether or not to decide to go into apoptosis. Also, during cell differentiation, stem cells need to decide when and how to move from a self-renewal state into a differentiated state. And each of these processes is tightly controlled by intracellular programs as well as extracellular signaling molecules, whose mechanisms are not yet fully understood. For example, we know that NANOC, OCT4, ZOX2 play a key role in maintaining stem cells in their self-renewal state. However, how this transcription factor regulate each other with other signaling genes dynamically to initiate the process of cell differentiation is not yet fully understood. So in order for us to model how the cell functions during this process, such as cell growth, cell proliferation, apoptosis, and cell differentiation, we need to examine their common properties. Firstly, cellular decision processes, they take time to completion. For example, in early embryonic development in mice, it takes about 1 week for stem cells to move from a self-renewal state into a differentiated state. Secondly, cellular decision processes, they operate in multiple steps. For example, for a stem cell to become a terminally differentiated cell, it needs to undergo through different stages of cell differentiation, and each of these stages can be activated by its own signal. Furthermore, cellular decision processes are controlled by complex signaling networks. For example, the Wnt signaling network, which plays a key role in the developmental process of tissues and organs in several multicellular organisms, have a complex mechanism occurring at the cell membrane, cytoplasm, as well as the nucleus. And once such a pathway is active, traces of gene expression can be observed downstream, corresponding to the various steps in the signaling pathway. So, for us to model a cellular decision process, we can model them by using hierarchies. So the figure shows you a cellular decision process comprising of 5 signaling steps: S1, S2, S3, S4, and S5. So S2, S3, S4, and S5 can only occur after S1 has occurred. For example, in the MAP kinase cascade, it's activated by RAS, and this further leads to the production of other important regulatory proteins that MYC. So in this example, we can say that RAS acts upstream of MYC. Similarly, in our example, we can say that S1 acts upstream of S2. So in a hierarchy, a node represents a single gene. And this is how we model the entire process. We intervene in the cell, and we look at what happens downstream at its target genes. And based on these expression changes, we are able to reconstruct the hierarchy. So formally, we can represent hierarchies as graphs, whereby the nodes are steps or genes controlling the various steps, and the edges indicate up-downstream relationship. With respect to up-downstream relationship, we expect the graph to be transitively closed. Since if S1 is upstream of S2 and S2 is upstream of S3, then by definition, S1 is upstream of S3. However, a key problem to gene perturbation screens is that the types, they occur downstream of the perturbed pathway. And these may not always show a direct influence of one pathway component to another. So this led to a class of models which was developed by Markovits and colleagues, known as the nested effects models. And in this model, we try to estimate or to reconstruct the hierarchy using the net structure of downstream effects. So if we have the downstream effects here having a nested structure, then we can state that the target genes of, say, S2, which are in green, are a subset of the target genes of, say, S1, if and only if S1 is upstream of S2. And this can be represented in the graph. I will not elaborate on this approach, but I would like to mention that our approach is based on this methodology. And this was presented in the ISMB conference in 2007 in Vienna. One drawback of this approach is that it cannot handle network dynamics. Because if S1 is upstream of S2 and S2 is upstream of S3, then logic will want S1 to be upstream of S3. But this does not imply that signal flows directly from S1 to S3. And if you take a closer look at the transitively closed triple, you see clearly that the direct edge combines with the indirect edge to form a feedforward loop. And Yuri Alon and colleagues have shown that these are a very frequent motif in signaling networks. And nature has made use of these feedforward loops in several organisms to cause time delays so that the cells can function properly. So we can also show that time delays might also serve as evidence of feedforward loops. The problem is that time delays between signaling events cannot be observed directly. They need to be estimated. Since we can only observe a signal propagation times from, let's say, an intervention S1 to some target readouts on a microarray, for example. And we want to estimate the signal propagation rate from S1 to S2. In general, given a certain hierarchy of signaling steps, we would like to estimate all the signal propagation rates on all the edges, given interventional data. However, in order to do this, you need to know which target genes are being controlled by which signaling genes. And dynamic steady effect tries to solve this problem. This figure simply shows you an easy to understand solution to the problem. On the left, we have a transitively closed triple comprising of 3 signaling steps, S1, S2, and S3. On the right, we have the time series data showing the effects on the target genes after intervention. A 1 simply means that the signal has reached the target genes before a certain time Tj. If you take a closer look at the hierarchy, you see clearly that we have 2 alternative paths to move from S1 to E3, which is the target gene of S3. And if you consider the longer path S1, S2, S3, and E3, you see that if we intervene at S1 and we look at E2, we see a 2-unit time delay signal for the signal to move from S1 to E2. However, if we intervene at S2 and we look at the time it reaches E2, it's just a 1-unit time delay. So giving us evidence of a 1-unit time delay between S1 and S2. Similarly, if we intervene at S2, E3, we have a 3-unit time delay. And if we intervene at S3 and we look at E3, we have just a 1-unit time delay. So this gives us evidence of a 2-unit time delay between S2 and S3. Looking at direct path from S1 to E3, if we intervene at S1 and look at E3, we have a 2-unit time delay. And if we intervene at S3 and look at E3, we have just 1-unit time delay. This gives us an evidence of just 1-unit time delay. And so this says that the direct edge is faster, and therefore it exists. So we are able to use estimated time delays to show the existence of feedforward loops. However, this is a simple scenario. In reality, gene expression is a stochastic process, And as I said, we don't usually know which target genes are being controlled by which signaling genes. And that is how the dynamic nested effect models works. So in principle, we model signaling as a stochastic process with exponentially distributed time delays. So given a hierarchy of signaling steps, we assume that each step is exponentially distributed with a certain expectation. So in practice, we actually model by the signal propagation rates. So we can have in a particular pathway, we can have an input signal to an output signal going through different steps. And so in this sense, we will be dealing with a convolution of exponentials. It could also be possible that since we are dealing with feedforward loops, there could exist also alternative signaling paths from an input signal to an output signal. And in the context of dynamic nested effect model, the first blocked signal wins in an AND gate situation. So as I said, we model rate signal propagation, and the actual times remain as random variables. If we assume a simple feedforward loop here, and we assume that the expected time spent on the edge or on the shortcut edge is ET1, and the time spent through Y is ET2, then the expected time for the signal to reach Z is definitely smaller than the expected time between the alternative paths. So in simulations or in practice, our model is going to predict, or we predict longer time delays than expected. And this is what we call the stochasticity effect. So to show how we estimate our model parameters, we basically estimate the signaling rates between all the edges. So associated with each edge or with each rate parameter, we fix a very large value, which we denote as lambda 0, corresponding to predictable time delay, and a very small rate value x, which simply means that the signal never arrives, or that there is no sensitive edge, or that there is no shortcut signaling. And since we all deal only with discrete parameters, we use the discrete sampling to estimate our model parameters, since we cannot explicitly calculate the joint distribution. And this makes use of a uniform prior for all the model parameters, except for a 0.5 prior on the x value, which kind of disfavors the transitive edges. So we give a higher so that if our model predicts a low posterior probability, then this is evidence that the edge is not present. We carried out several simulation studies, and in one of them, we used this known graph comprising of 6 signaling steps. We generated data assuming a finite time delays on the transitive edges, and we assume that the transitive edges are non-existent or there is no signal. We generated data based on 3 noise levels: low noise, high noise, and high noise. And we applied our dynamic model. And the posterior distributions are summarized in the heat map shown here. So the rows represent the edges, the columns represent expected time delays. And the last column represents the probability mass on the x parameter. The first column represents probability mass on the lambda 0 parameter. A light gray represents a very high probability. A dark gray represents a very low probability. So if our model predicts a finite time delay, then there is evidence for the existence of that edge. And if x has a high probability mass, then there is evidence that the edge doesn't exist. So here we have certain red edges in red corresponding to the transitive edges. So our model, if you look at x, it has high probability mass on all these edges. Therefore, our model is able to detect or to predict that these edges are non-existent. Now we turn to a real data scenario. We use data from the Lemishka's lab. And it has been shown that NANOC, OCT4, and ISRB, TBX3, and TCL1 are all downregulated upon cell differentiation. So in order for us to model how dynamically how the signal mechanism between these regulators, we carried out RNA knockdowns were carried out for the various genes over a period of 7 days, giving us measurements or gene expression measurements for 8 time points, including the baseline. So upon careful discretization of the data using both the positive and the negative controls, we have this nested structure of downstream effects at the last time point, especially corresponding to the NANOC, ZOX4, OCT4, and TCL1. You could see a clear cascade between these 4 transcription factors. However, we don't know how TBX3 and ESRB interact with the cascade, or whether there even exist feedforward loops or not. We use the Markowitz algorithm to estimate our hierarchy, and then we applied our dynamic model on the time series data. And the result is summarized in this heat map. So as I mentioned earlier, a light gray corresponds to a high probability, posterior probability. A dark gray corresponds to a low posterior probability. Our model is able to remove 3 transitive edges. And also, you could see that there are kind of like very fast underlying processes. They're kind of like also slower underlying processes and even longer underlying processes in the whole transition from undifferentiated cell to cell differentiation. So we can summarize information on such a heat map into this non-transitive graph showing the direction of signal flow as well as the qualitative propagation on the edges. We see clearly that there are forward loops which kind of dominate in the network. And these feedforward loops kind of stabilize the differentiated signals with respect to self-renewal by filtering out random fluctuations as a random noise. Also, we see that NANOC plays a key role in cell differentiation. And one possible role of the remaining signaling gene is probably making cell differentiation a one-way process. A reversal will probably lead to a serious cancer situation. To conclude this presentation, we have seen that the dynamic effect models can be used to model the dynamics of networks from from interventional microarray data, time series microarray data. Also, we have seen that they can infer feedforward loops from estimated time delays, as well as capture the stochastic nature of signal processes. I would like to acknowledge all those who helped in this work. Special thanks goes to Rainer Spang, who is actually my supervisor and head of the group. I would also like to thank Mohamed Sadeh, Peter Offner, Juby Jacob, Akim Tresh, Marcel Vlad, and Florian Markowitz. I would also like to thank your attention.
Len, do you have time for questions? Not many questions. So will you able, I was thinking in terms of stochasticity, consistency with the number of molecules are known. To use this as input. And in your system it's more complicated because it's an eukaryote complicated system, but you go to the bacterial system with the number of molecules. It's a small known number.
You mean if we can go to a lower level like molecules.
You can take, go to the stream of the term of stochasticity and think in terms of number of molecules. There are 20 molecules doing
I
something.
see.
Because things may happen or not happen because there may be or not enough molecules in these cells to do them.
Yeah, but we are dealing with interventional data, so which means that, of course, intervene at more than one molecule is no problem. So I think the approach can still handle more than one molecule as long as you are able to carry out the experiments.
Well, there are experimental data for this.
Data. Yeah, but this approach is mainly for interventional data. So which means that somehow you need to intervene in the cell, and then you can only have indirect measurements. So we're kind of like trying to reconstruct networks based on indirect measurements. Yeah.
Yeah, no, I understand that, but you can imagine a situation in which you take this data from an experiment, but then you know that in the system normally 20 molecules this time. So there's a constraint to your the system.
Of course.
Oh, think on that.
Okay.
Nice work. I was wondering, regarding the application, if next steps involved also just trying to capture more of the stem cell differentiation process by adding time points, and if so, do you have any intuition as to how many more time points one would want to study?
You mean if we can add more time points to the data?
Correct.
Okay, then I think that might just help to be more robust in terms of prediction, but our assumption in this model is one-state transition. So we kind of like model processes that are just in one-state transition, like the self-renewal into differentiation. So probably, I mean, if you have more time series or more repetitions at a particular time point, that will be useful. But still, I believe that this method is going to work best if you actually have the times at which things happen. If your data has the information, no problem. So if you can improve or increase the number of measurements and you still have the right signals at the right times, then it's okay.
Thank you.
Yeah, this was an interesting talk. I was wondering if you had, so now at the end of this process, right, you get a sort of, arrow diagram with this sort of, qualitative kinetics, right? So can you use that result to go back and do like forward simulation of any kind, like just a simple forward simulation? Because then you might be able to sort of come up with some hypotheses of why you see like particular differences in kinetics, right? Like what states that lets you avoid.
Yeah, I think that is a good point. Yeah, it's a good point. Normally, the problem is that since we are actually working on the modeling aspect, and usually the getting hold of such a data is not always easy. So usually, it's like you really need to also do a follow-up study for this year, which is okay.
Please.
Yeah, hi, great talk. I love the addition of the temporal aspect to the nested effects model. Can you comment on how the running time of the algorithm and how it might scale to a larger system?
That is an interesting question, actually. And since we are this is just GIP sampling, which means you basically don't use any approximations, you sample everything. Of course, for small systems, it's okay. But for larger systems, it would definitely going to take a longer time. So for smaller systems, of course, it can take probably days. But for larger systems, of course, it would.
If I understand, you're running Florian's method as part of your code, which
no. For this method, we already assume that we have the hierarchy.
Okay.
That is why we focused last time point, because we are dealing with one-state transitions. So we can actually get the hierarchy at the last time point, because then you have all the accumulation of effects. But then the problem is that you still need to decide through the dynamics which process occurred faster or earlier than the others. So you kind of like, this approach assumes that you already have the hierarchy.
Well, there are more questions. Thank you.